Those green threads are nerve fibers. The blue mass they run through is a lung tumor in a mouse, imaged whole after the tissue was made transparent. The fibers were labeled at the far end in a knot of nerve cell bodies at the base of the skull, and then traced down into the chest. Every green line you can see grew into that tumor from up there. The tumor did not build that nerve. It pulled an existing one in. Which raises the question this whole result turns on. What is traveling along that line, and which way is it going? Signals go up from the tumor to the brainstem, and something comes back down. Not a growth signal for the cancer, but an instruction that quiets the immune cells that would otherwise be killing it. Cut that loop anywhere along its length, and the tumors get smaller, because the immune attack switches back on. That is a target that is neither the cancer cell nor the T cell. It is the wiring between them, and the far end of that wiring is a receptor that cheap, common drugs already bind. The work was published in Nature in February 2026 by Haohan Wei and Chuyu Yu across Chengcheng Jin's cancer biology lab at the University of Pennsylvania and Rui Cheng's neuroscience lab at Yale. The mice carry a lung adenocarcinoma driven by two of the commonest changes in the human disease, an activating KRAS mutation and the loss of p53. Tumors start in the alveoli, the air sacs at the ends of the airways. To test whether the tumor was recruiting nerves, the team grew vagal sensory neurons in a dish and bathed them in fluid drained from real tumor explants. Neurons in plain medium put out short, stubby processes. In fluid, from healthy lung, much the same. In tumor fluid, they threw out long, branching neurites, and heating the fluid first took most of that away, so whatever does the recruiting is a protein. Not every vagal neuron answers. The vagus is a bundle of molecularly distinct sensory lines, each reporting on something different. The ones that grew into these tumors carry two markers, NPY2R and TRP51, and about 9 in 10 of them carry both. In a healthy lung, that population sits in the alveoli and reports on breathing. A different vagal population, marked by P2RY1, wires to the airways instead, and it is absent from the tumors, and destroying it does nothing to tumor burden. Destroy the sensory neurons of the spinal ganglia instead, and again, nothing. General nerve damage is not what helps these mice. So they cut the line 4 separate ways. A genetic switch that makes only those cells vulnerable to diphtheria toxin. A chemical injected into the ganglion to burn out the TRP51 cells. A designer receptor that lets an injected drug silence only the lung-projecting ones without killing anything. And at the far end, deleting the receptor the signal lands on. Every one of them shrank the tumors. It held in the slower model, too, where the cancer arises in the lung over months instead of being transplanted in. The mice lived longer, lost less weight, and went back to building nests. Now follow the signal. Switch those neurons on in a healthy mouse, and 2 regions of the brainstem light up with FOS, a marker left behind by recent firing. First, the nucleus of the solitary tract, where sensory information from the internal organs arrives. Then, the rostral ventrolateral medulla, one of the places the brain sets sympathetic tone. Tumor-bearing mice show both regions running hot on their own, and ablating the sensory neurons brings both back toward healthy levels. It works from the other end, too. Silence the neurons in that medulla region directly, touching nothing in the lung at all, and the lung tumors shrink. The return leg is sympathetic. Firing rises in the sympathetic ganglia beside the spine that supply the chest, and their fibers are physically there in the tumor bed. Cut the sensory input, and the sympathetic fibers around each tumor fall about 5-fold, and the noradrenaline in that lung tissue falls with them. The timing rules out the obvious objection. At day 18, before the tumors differ in size at all, the brainstem activity and the fiber counts have already dropped. The quieter circuit came first. Which leaves the last question. What does the noradrenaline do down there? Put it on these cancer cells in a dish for 3 days, and they grow the same as before. Put it on killer T cells, attacking those cancer cells, and the killing carries on unchanged. The cancer cell is barely listening. The cell that is listening is the alveolar macrophage, the resident immune cell patrolling the air sacs, and the lung cell loaded with the beta-2 adrenergic receptor. Noradrenaline pushes it into an arginase-high state and strips down the machinery it uses to present targets. The T cells around it stop making interferon-gamma and TNF. They are still there. They have gone quiet. 2 deletion experiments pin down the order. Remove the CD4 and CD8 T cells, and cutting the nerve stops helping. The benefit was always the T cells doing the work. Remove the alveolar macrophages, and cutting the nerve stops helping either way. The nerve acts on the macrophage. The macrophage acts on the T cell. Then they ran it forwards. Take mice whose sensory nerves have already been cut, and put the far end of the signal back chemically, a nebulized beta-2 agonist, Salbutamol, 45 minutes a day. Tumor burden came back to roughly where it had been before the nerve was cut, and the T-cell response fell again. Cut the wire and the tumor shrinks. Supply the missing chemical and it grows back. That is the step that turns this from a correlation into a lever. One thing it is not. A sustained daily aerosol in a mouse is not evidence that asthma inhalers cause lung cancer in people, and a Korean study of 63,000 inhaler users found no difference in lung cancer incidence between regimens. So how much of this is true in a person? Honestly, less than you would like. They ran a nerve gene signature across 518 human lung adenocarcinomas, and a high score tracked with worse survival and with fewer CD8 T cells inside the tumor. A correlation in bulk tissue, not a traced circuit. The receptor at the far end is the most abundant adrenergic receptor in human lung cancer, which is the encouraging part, because beta blockers are old and cheap. Several retrospective series do report better outcomes in patients already taking them, nearly 5,000 in the largest. But the record is not clean. One large analysis found higher mortality among beta blocker users, and a pooled meta-analysis found nothing. The only prospective lung trial pairing a beta blocker with immunotherapy has 6 people enrolled and no results. The part I keep coming back to is that the circuit was already there. A sensory line from the alveoli up to the brainstem and a sympathetic line back down is ordinary lung physiology. It is how breathing and blood pressure get matched to what the lung is doing. The tumor invented nothing. It grew into an existing control loop and let the body's own regulation do the suppressing for it, which is also why cutting the vagus is not a therapy. The same system restrains dangerous inflammation elsewhere, and you would be trading one problem for another. Anything useful has to be far narrower. One receptor on one cell type in one organ. And this is early. 3 months later, a group at the Crick Institute in London reported a second nerve mechanism in lung cancer, a local one running through a different neuropeptide, and blocking that same neuropeptide did nothing at all in these experiments. 2 labs, 1 organ, 2 different nerves, and nobody yet knows how many of these lines a tumor is running.